My goal was to explain where glucose 6-phosphate comes from, and also to explain the glucose 6-phosphatase enzyme and how you would test its functionality on a liver biopsy assay. So glucose actually comes in the cell. It usually comes through a GLUT transporter, but the cell is slightly permeable to glucose, not very much at all actually. So you get down past 10 to the -4th and you have quite a bit of trouble getting across the membrane, but it is slightly more permeable than an ion. This is important because some glucose can just go into the cell, very, very small amounts, and whenever it gets into the cell from extracellular fluid, you have it immediately being transformed into glucose 6-phosphate by glucokinase. So glucokinase uses ATP and adds an ATP to glucose, and that actually gives it a negative charge and makes it extremely hard. So it's not just a small molecule that is charged, it's this large molecule that's charged, so it would have a even lower permeability than these ions here listed. And that does two things. So the first thing it does is it traps it in the cell, so it traps it. And the second thing that it does is it reduces the cellular concentration of glucose. So if the cellular concentration of glucose goes down, that means any type of diffusion into the cell would increase. So that increases the cell's ability to uptake more glucose. And that's because the concentration gradient that moves glucose into the cell is only dependent on the amount of glucose in the cell. It does not, if glucose gets changed into something else, then that takes the concentration of glucose in the cell downward and allows the concentration gradient to continue crossing the membrane. And rather than the membrane, typically it's going to be a GLUT transporter. Now it's important to remember that there's many types of GLUT transporters, and they all have a slightly different affinity for glucose, and they all have a little bit of a different purpose or functionality and where they're located and what kind of histology they're located in. So the first place where we get glucose, the first place where glucose 6-phosphate comes from is by importing extracellular glucose. So I'm going to put extra glucose, and glucokinase transforms it into glucose 6-phosphate. Now it's also important to know that some cells use hexokinase, and some cells use a very specific hexokinase called glucokinase. So hexokinase is kind of non-specific and can work on many different 6-carbon sugars, whereas glucokinase can only work on glucose. Now, specifically, the liver loves glucokinase, and that's because the Michaelis constant is different. So it's going to have a lower affinity for glucose than a hexokinase would, and that has some interesting physiological consequences. Now, the second place where you can get glucose 6-phosphate is by the phosphorylasis of glycogen. So glycogen gets broken down by an enzyme called Glycogenphosphorylase. So Glycogenphosphorylase. Now, remember when we're talking about enzymes, we've covered kinases. So a kinase takes a phosphate group, usually from another organic phosphate, and transfers it to another organic compound. So this is from organic to organic. A phosphorylase, which I'm not going to rewrite that out, usually takes an inorganic phosphate and adds it to an organic molecule, making a brand new organic phosphate. And then you have phosphorylase, a phosphoratase. If it ends in TASE, then typically it's removing. So it would remove, we'll just put remove a phosphate group from an organic compound. So in this case, we're taking this glucose, we'll just draw this out and pretend that each one of these circles is a sugar, and they're linked by a glycosidic bond in between them. And a phosphorylase would take an inorganic phosphate and add it, and it would remove H2O. And that would actually cause the compound to break apart. And so glycogen phosphorylase can take one at a time from the terminal glucose monomer and break it off of the glycogen. Now, once it gets down to this point where it's branched, the branching portion causes you need a different enzyme. So you actually need something called debranching enzyme at this junction right here. So debranching enzyme would allow that to be broken off. But typically we're looking at glycogen phosphorylase. That creates glucose 1-phosphate. Then you could use, and let me make that a little bit smaller. And once you have glucose 1-phosphate, phosphoglucomutase will turn it into glucose 6-phosphate. So that's the second source of glucose 6-phosphate in the cell. The third source of glucose 6-phosphate in the cell is gluconeogenesis. So gluconeogenesis, and this is when whenever glucose typically goes into a cell for metabolism, it breaks it down to pyruvate in a process called glycolysis. So glycolysis, and this process is irreversible at 3 key points. So hexokinase or glucokinase, it will only go from glucose to glucose 6-phosphate. So that's when it first enters the cell. So that's an irreversible step under physiological conditions. Then fructose 6-phosphate, acting on by phosphofructokinase to produce fructose 1,6-bisphosphate, is also irreversible. And then the creation of pyruvate by pyruvate kinase is also irreversible. But using a different pathway, we can actually go from pyruvate or any one of these intermediates back up to glucose. And that's one of the sources of glucose 6-phosphate, is the reversal of this. So what happens here is pyruvate will have a carbonate, so CO3 negative. So carbonate will be broken apart, and CO2 will be added to pyruvate to create oxaloacetate. Okay, I know that this is a little bit hard to read, so I'm going to zoom in down here. And it's a little bit blurry, but just bear with me and hopefully you'll be able to understand. So pyruvate here is acted on by pyruvate carboxylase. So this pyruvate carboxylase turns it into oxaloacetate. Now, this step isn't shown, but oxaloacetate is formed inside the mitochondria, and so it goes through the malate aspartate shuttle, and so it gets converted into malate and then back to oxaloacetate outside of the mitochondria, and where phosphoenolpyruvate carboxykinase turns it back into phosphoenolpyruvate. Now, the rest of these steps from phosphoenolpyruvate are actually right here, all the way up to fructose 1,6-bisphosphate is reversible. So you just increase the concentration of PEP, phosphoenolpyruvate, and it will drive the reaction up to fructose 1,6-bisphosphate. And then once you get to fructose 1,6-bisphosphate, instead of having, and this is actually, well, just to be sure there's no confusion, let me go ahead and show you this. I found, whenever I downloaded this picture, I found an error on it. So this says glucose 6-phosphatase, which is incorrect. Glucose 6-phosphatase is what acts right here. This should be fructose 1,6-bisphosphatase. So remember, a phosphatase removes a phosphate group from an organic compound. So it's removing one of the phosphates to make fructose 6-phosphate. And I said, of course, remember, hexose kinase acts on any 6 carbon sugar. But this specific hexokinase we said was phosphofructokinase. So phosphofructokinase is the one driving this reaction. Now, once we get up to glucose 6-phosphate, that's where, at that point, we have our third source of glucose 6-phosphate. But remember, that can also be converted back to glucose through glucose 6-phosphatase. So a phosphatase removes another phosphate and you get glucose. So that's the three sources of glucose 6-phosphate. You have from glucose, from glycogenolysis or glycogenolysis, however you want to say it, genolysis, and from gluconeogenesis, And those are the steps. I just explained them all to you. Now, whenever we want to take glucose 6-phosphate and turn it into glucose, so this happens only in the liver and in the kidneys, and it can happen in a few other cells, but to a very, very small amount. So for physiological purposes, taking glucose 6-phosphate and turning it into glucose, we're just going to say it happens in the liver, because that's where it's physiologically important. And that's done by an enzyme called glucose 6-phosphatase, so it's a glucose 6-phosphatase. G6Ptase is the abbreviation for it. But the way that's controlled is by where glucose 6-phosphatase is located. Now, glucose 6-phosphatase is the last step in getting glucose 6-phosphate into glucose so that it can go and power the rest of the cells. And now it's inside of the endoplasmic reticulum. The glucose 6-phosphate gets in there by a multi-subunit enzyme complex. So it's called glucose 6-phosphate translocase, which is a 3-subunit enzyme. And its subunits are glucose 6-phosphate translocase 1, glucose 6-phosphate translocase 2, and translocase 3, all just labeled as T1, T2, and T3. And so the glucose 6-phosphate would travel in here. It gets acted on by the phosphatase, and then it travels out through T3. The inorganic phosphate that's released can travel out through T2. So this T2 is kind of important because it's bidirectional, whereas these two are unidirectional, and not only can inorganic phosphates travel through it, but pyrophosphates can also travel bidirectionally through this enzyme channel. So if there's a problem with converting glucose 6-phosphate into glucose, the problem could be one of two things. It could either be that the glucose 6-phosphatase enzyme is broken, or the transporter is broken, or defective, we would say. So here we have the cell membrane, so glucose will come into the cell, or if they're under low glucose, we can ship glucose out of the cell. But this is the membrane of the endoplasmic reticulum. So we're inside of the endoplasmic reticulum, and we have this glucose 6-phosphatase. So if we have a lot of glucose 6-phosphate, it can actually be transported into the endoplasmic reticulum through this. There's a complex called the glucose 6-phosphate translocase. Glucose 6-phosphate translocase. And that's a 3-subunit enzyme. The 3 subunits are translocase 1, translocase 2, and translocase 3. When there's a high amount of glucose 6-phosphate, it will move across the translocase and into the ER, where glucose 6-phosphatase will convert it back to glucose. And it removes the phosphate, and we have an inorganic phosphate that can move out of T2 and a glucose that can move out of T3. Now, it's important to note that T1 is unidirectional. You can only move in. T3 is unidirectional, you can only move out. Now T2 can move phosphate and pyrophosphate out or in, so they just move along their concentration gradient. So why is it important for pyrophosphate? So pyrophosphate, it's basically 2 phosphates bound together. And the reason I'm letting you know this is because glucose 6-phosphatase is pretty non-specific in that it will remove phosphate groups from more than just glucose 6-phosphate. So if pyrophosphate came into the ER, it could act on pyrophosphate, breaking off an organic phosphate molecule and sending it out as well. So glucose 6-phosphatase is also a pyrophosphatase. Now, if you haven't already picked up on it, I'm looking towards the diagnosis of glycogen storage diseases. So if you have a glycogen storage disease, there's multiple types. But glycogen storage disease 1, GSD1, also called von Gerk's disease, so GSD1, has two subtypes, and it's the same phenotype. So glucose 6-phosphate cannot be converted into glucose. Now, this only happens in the liver for physiological importance. So when you diagnose this in the past, nowadays we could just do a DNA sequence, or we could just test the genes and we could have a positive diagnosis. But in the past, a liver biopsy would be taken because it would be relatively important to know if you had glycogen storage disease 1A or glycogen storage disease 1B. Both of them end up with the same phenotype. So the phenotype is that glucose 6-phosphate is not converted into glucose, and because it's not converted into glucose, you're not getting glucose sent out into the blood, and you end up being hypoglycemic. Now, so type 1A means that the enzyme is defective. Type 1B means that the transporter is defective. And so if the transporter is defective, you can't convert it, and because it won't cross into the ER and it won't be converted. If the enzyme is defective, it'll cross into the ER, but the enzyme won't do anything to it, so it won't get converted. So you get the same phenotype. So you biopsy the liver, so you got your liver here. You take a biopsy of it, and out of that tissue that you've removed, you put it in a tube and you homogenize it. When you homogenize cells, what happens to the endoplasmic reticulum is it breaks up into small little droplets that we call microsomes. So all of the endomembrane system essentially becomes microsomes, and most of that is the endoplasmic reticulum. So whenever you spin that at 1,000 times gravity, you get a little what's called the nuclear pellet. And the nuclear pellet's down here. Then you pour off the supernatant and you spin it again at 20,000 Gs. You get the mitochondrial pellet. So these are all mitochondria. Then you pour off the supernatant and spin it again, and at 150,000 times gravity, and you get the microsomal pellet. So let's just imagine that we've got this microsomal pellet. So we decide to split it up into 4 different test tubes to assay it. So we get rid of we have our microsomal pellet right here, and we've got all the supernatant on top of it. So all this liquid, we're going to pour off the supernatant, and then we're going to reconstitute it with some other liquid medium to basically break this up into the liquid, and then we'll pour a little bit into each one of these tubes. And in these 2 tubes we add no detergent, but in these we add detergent. So plus detergent, and then so we've got our microsomal fractions here, and into that microsomal fraction we're going to add glucose 6-phosphate to that one, and we're going to add pyrophosphate to this one. Then we're going to add glucose 6-phosphate to this one and pyrophosphate to this one. Now in this case, let's talk about what we find. Whenever the T1 transporter is messed up, you'll get glucose going in here, but T1 will not transport it into the microsomes, so you get no inorganic phosphate being released. Pyrophosphate, however, will cross the T2, and it will be acted on by glucose 6-phosphatase, and it will release inorganic phosphate. So the inorganic phosphate we get from this one is 0. The inorganic phosphate we get from this one we'll just say is 300 milliequivalents. And then whenever we break up that membrane, all of a sudden the glucose 6-phosphate is free to access that enzyme that it couldn't access. So in this case we get our inorganic phosphate level is 250, and in this one it's 275. So we're just using the pyrophosphate in this case as a control to make sure that we didn't do anything else wrong. And this assay would indicate that our T1 transporter is messed up. So that would indicate Glycogen Storage Disease 1B. Now, as I said before, this is a slightly historical method, so in modern days we would just use DNA sequencing or do some type of genome analysis, and we would be able to determine, oh, there's a problem with T1 gene, or there's a problem with the glucose 6-phosphatase gene, and that would be diagnostic. You wouldn't have to do this assay or the biopsy, so it's less invasive. You see, the reason for the detergent is to just break open the membrane, and any enzymes that are down here can then be released to their substrate, which would be glucose 6-phosphate in this case. And so again, some people had questions, why is detergent added to that assay? Well, because if you have a membrane, so this right here, instead of saying cell membrane, let's imagine it says endoplasmic reticulum membrane. Detergent is basically a bunch of micelles, and the micelles will go in and break up this lipid bilayer and incorporate pieces of the lipid bilayers into the micelle, and therefore it tears it open and allows access to the outside material. In summary, there's three sources of glucose 6-phosphate, so glucose 6-phosphate, and that is from glucose through a hexokinase, and from glycogenolysis or glycogenolysis, so glycogenolysis, and then from gluconeogenesis, so from gluconeogenesis. Whenever that glucose 6-phosphate cannot be converted back into glucose by only the liver or the kidneys, but we'll just say by only the liver, then it's usually because there's a problem with the T1, the glucose 6-phosphate translocase, the translocase 1 to be specific, or it's because there's a problem with the phosphatase enzyme. Both of those respectively are Glycogen Storage Disease 1B for the translocase and Glycogen Storage Disease 1A for the phosphatase. And I believe collectively they are known as von Gerk's disease. So von Gerk. And I may have spelled that wrong, but von Gerk's disease is GSD1 disease.